Hartnup disease is a rare inherited disorder that exemplifies the systemic impact of disrupted amino acid transport. It bridges molecular pathology with distinctive clinical features, making it an essential topic in metabolic medicine. Hartnup disease is a rare autosomal recessive disorder caused by mutations in the SLC6A19 gene, which encodes a sodium-dependent neutral amino acid transporter. This transporter is primarily expressed in the epithelial cells of the renal proximal tubule and the small intestine. Its main role? Absorbing neutral amino acids like tryptophan, a precursor for niacin, serotonin, and melatonin. When the SLC6A19 gene is defective, neutral amino acids are poorly absorbed from the gut and excessively excreted in the urine. This results in systemic tryptophan deficiency, impairing niacin synthesis, and leading to pellagra-like symptoms: photosensitive dermatitis, cerebellar ataxia, and emotional instability. Clinically, Hartnup disease often presents in childhood, frequently triggered by illness, poor nutrition, or sunlight. Many cases are asymptomatic and only discovered through aminoaciduria on urinalysis. Treatment is typically supportive. High-protein diets can compensate for poor intestinal absorption. Niacin supplementation often resolves neurological and dermatological symptoms. Avoiding excessive sun exposure is also key. Emerging research is exploring targeted amino acid therapies and gene expression modulators that may one day correct the underlying transport defect. These therapies remain in the experimental phase, but they hold promise for more precise, genotype-based intervention. Hartnup disease is a reminder of how metabolic pathways and transport systems underpin broader clinical syndromes. In our next segment, we'll tackle another amino acid disorder, one with a vastly different pathogenesis and prognosis. Don't miss it.